Awesome. Thank you, Shelley. So thank you everyone for coming to my defense. My name is Alicia Iwanicki and I am a master's student at the Lab of Atmospheric Research at WSU. So the topic of my presentation today is measuring continuous nitrous oxide emissions with an automated flux chamber system in an agricultural field from a cover crop and a winter wheat system. So in the first part of this presentation, I'll go over some background information on the nitrogen cycle that will help in understanding my data and the results and the motivation behind doing this study. Then I will discuss the specific questions that we aimed to answer with this research. I will then explain the experimental design and methods that we used. Then I will cover the data and findings, and I will end by synthesizing my conclusions. So why do we care about nitrous oxide? Nitrous oxide, or N2O, is a potent greenhouse gas with nearly 300 times the global warming potential of CO2, and it is the main contributor of stratospheric ozone depletion. N2O atmospheric concentrations have steadily increased since about 1750 due to human activity, and is currently about 20% higher than pre-industrial levels. And concentrations are continuing to rise approximately linearly at a rate of about 0.3% per year. Nitrogen fertilizer application to croplands contribute the vast majority of global N2O emissions, about 78% in the US. And the most recent FAO World Fertilizer Trends Report estimates that in 2020, the world demand for nitrogen fertilizer is around 109 teragrams of nitrogen per year, which is 109 billion kilograms. And to put that into context, in 1950, at the start of the economic boom, usage was only at around 3 to 4 teragrams per year. And currently demand is increasing at a yearly rate of about 1.5%. So to understand how N2O is formed in the soil, we need to understand the basics of the terrestrial nitrogen cycle. So nitrogen is an element that we all need to survive. Plants use it to grow, and humans and animals consume it to acquire it. 78% of the global nitrogen is in the atmosphere as gaseous N2, and it is unavailable to plants in this form because it is held together by a strong triple bond. So first, this N2 needs to be fixed into a reactive nitrogen form so that it can combine with other elements to create compounds like ammonium, NH4, or nitrate, NO3. And these are the two forms that plants acquire nitrogen. And NO3 is the preferred form because it is more mobile. So this figure here shows the main sources and sinks of reactive nitrogen in the global terrestrial pool via anthropogenic and natural nitrogen fixation. So the arrows, or the sizes of the arrows, show the relative pathway magnitudes. And I've also left off error values just to keep it simple. And all of these units are in teragrams of nitrogen per year. So where 1 teragram is 1 billion kilograms of nitrogen. So here we have the main anthropogenic pathways, how reactive nitrogen enters the terrestrial pool. And nitrogen fertilizer production is predominantly done currently via the Haber-Bosch process, which fixes about 2 times more atmospheric nitrogen than natural terrestrial sources yearly. So we have 63 compared to 110. So I also have legumes here, and you can see that legumes add an additional 60 teragrams, and I'll cover more on this on the next slide. The main removal of reactive nitrogen from terrestrial systems is believed to be denitrification via gaseous nitrogen emissions, and this right here is ammonia volatilization. So I will talk more about the denitrification process on the next slide. Of the 13 teragrams of nitrogen emitted as N2O per year, about 2.1 teragrams is from agricultural soils. So up here, I've shown those main pathways that reactive nitrogen enters the terrestrial system again. The green bubbles show the natural ways, and the gray show anthropogenic. And so I just want to come back to the legumes here. So legumes such as peas and beans have special bacteria called rhizobia that live in the nodules attached to the roots of the plants. and they can fix atmospheric nitrogen into ammonium. And then the ammonium diffuses out of the bacteria and into the plant root for uptake. So they don't immediately contribute ammonium to the soil for other plants to use. They only do so once they die and decompose, and it's added via subsequent mineralization. And mineralization is the oxidation or decomposition of chemical compounds in organic matter, such as dead plants and animals, which releases those nutrients into soluble inorganic forms of ammonium, making it available to plants. And finally, we have chemical nitrogen fertilizer. So remember when I said that plants can uptake either ammonium or nitrate? Well, these are the two forms of nitrogen fertilizer that are typically applied to crops. And like I had said, anthropogenic activities have more than doubled the amount of reactive nitrogen via, or in the global nitrogen cycle in the last 50 years via nitrogen fertilization. And the issue with this is that typically less than half of the fertilizer that is applied to crops is actually absorbed by the plants. So with all that extra reactive nitrogen, these microbial processes right here get amplified under the right conditions. So these are the main N2O producing soil processes: nitrification and denitrification. And the microbial communities that are involved in each of these processes is different. So nitrification is an aerobic process. So oxygen is present in the soil, where autotrophic nitrifier bacteria oxidize ammonium or ammonia into nitrite and then into nitrate. And N2O is released through the decomposition of this reactive intermediate right here. For this process to occur, water-filled pore space must be around 30 to 60%, so soil is not saturated. Denitrification, on the other hand, is an anaerobic process that occurs under saturated soil conditions, where water-filled pore space is above 60%. And here, heterotrophic denitrifying bacteria use the nitrate for their respiration in the place of oxygen, reducing it to nitrite, nitric oxide, nitrous oxide, and finally back to molecular N2. And this pathway generally does not go to full completion, and different denitrifying bacteria are responsible for each step. So basically what you need to remember is that saturated soil conditions means denitrification, and aerated soil means nitrification. But if it's too dry, then nitrification, or neither process, will occur. So these two processes are coupled in natural settings because the denitrifying bacteria are dependent on nitrification for the production of nitrate substrate. But the addition of nitrate fertilizer essentially decouples these processes, and N2O production via denitrification increases substantially when the soil is waterlogged. So soil-based N2O emissions are produced through nitrification and denitrification, and they are correlated to changing soil conditions as a result of environmental and anthropogenic drivers. So I've touched on most of these already. Soil drying and re-wetting, more commonly known as the Birch effect, as well as freeze-thaw, are more event-based cycles that have more recently been observed to be major drivers of N2O production in soils. With soil re-wetting after a long dry period, whether from rainfall or irrigation, the addition of water can rapidly increase mineralization rates and thereby the production of substrate for nitrifying bacteria, which then creates substrate for denitrifying bacteria, and overall N2O emissions increase. Freeze-thaw events occur during the non-growing season when either trapped N2O produced under frozen soil surface layers is released, or when frozen layers or snow thaw and saturate the soil, driving denitrification. And N2O loss from agricultural soils in the winter and spring have been observed to account for about 60 to 80% of the annual N2O emissions from several studies in the literature. So ultimately, nitrogen substrate availability is the main driver, whether through natural mineralization processes or through additions of synthetic fertilizer. And all of these influences affect microbial populations and diversity, which are as heterogeneous as the soil itself. And this results in the reaction rates of the soil biogeochemical processes to vary both spatially and temporally. And these are defined as hotspots and hot moments. So hotspots are discrete areas of enhanced N2O emissions relative to the surrounding soil. And hot moments are short periods of time with disproportionately high N2O emission rates. And prolonged hot moments are commonly referred to as pulses. So two anthropogenic influences on N2O that I haven't quite talked about yet are no-till management and diversifying crop rotations with cover crops, for example. In the literature, both frequently have been shown to increase N2O emissions, and our study site is a no-till site, and we also simulate a crop rotation. These higher N2O emissions are usually explained by a higher soil organic matter in the top surface layers of the soil, resulting in increased soil density, moisture, and mineralization, creating optimal conditions for microbial activity and thereby N2O emissions. So the overarching goal of this study was to measure continuous N2O fluxes from a small scale plot on an agricultural field under different vegetation types and management practices. And we did this by implementing two successive cropping systems, a cover crop system and a winter wheat system. And then we analyzed I analyzed the environmental and anthropogenic influences on the spatiotemporal variability of N2O fluxes from each. So up until now, I've talked about how N2O is emitted from soil due to microbial processes, but how do we measure the rate of N2O concentration change from soil, commonly referred to as flux? So we are using automated static chambers to make these measurements. To do this, a PVC cylindrical collar is inserted into the soil surface, and a chamber base plate is placed over top, and the chamber dome closes down over top of it. Vents in the chamber ensure that there's no pressure differential created inside the chamber, so we're not actually pulling soil gas out of the soil. The closed chamber then captures N2O that diffuses up through the soil pores from the sites of microbial N2O production. And following Fick's first law, a decrease in concentration gradient between the soil to chamber air over time will inherently result in a nonlinear increase in chamber gas concentration. So this makes nonlinear regression model analysis more appropriate than the traditionally used linear model, since linear regression will almost always result in inaccurate or underestimated fluxes because it assumes that the rate of concentration change is constant. So to calculate the fluxes from all of the chamber measurements that we made, we used a flux calculation software that calculates both the linear and exponential flux from the concentration curve data. So you can see an example of such a chamber measurement right here, showing both linear and exponential regression. So I calculated fluxes mainly using exponential nonlinear regression, unless that model could not converge to the concentration curve, in which case I defaulted to linear. So our study was conducted at the Palouse Conservation Field Station, or PCFS, and it is a USDA Agricultural Research Service study farm located near Pullman, Washington. And I've shown it here outlined in blue. And it has been used to conduct various soil, water, and crop experiments since about 1930. The climate here is classified as warm summer Mediterranean, and in 2019, we recorded an annual precipitation of 450 millimeters with an average annual temperature of 9 degrees Celsius at PCFS, which is typical of this region. And as everyone knows, the majority of the precipitation falls or occurs in the fall, winter, and spring, as rain or snowfall, and the summers are predominantly dry and temperate. So compared to CO2, which is usually emitted from soils in high concentrations, N2O soil emissions are difficult to quantify due to their small magnitude and high spatial and temporal variability. So to see how much N2O emissions varied even over a small area of soil in response to various drivers that I had mentioned earlier, we selected a 12.5 meters squared study plot in an agricultural field at PCFS and divided it up into 16 microplots as shown in the diagram right here, and as well as in the photos. We then applied various crop and fertilizer treatments to the microplots, and I'll explain all of that later. So to measure the N2O fluxes from the soil, 2 independent instrument systems were set up, each including 8 automated flux chambers, so 16 in total, 1 flux chamber per microplot. And these are all connected to a multiplexer, which cycles through each chamber one by one, slowly pulling up the sample gas from the chamber for a 12-minute measurement period. And the sample gas then cycles through a CO2 analyzer and then through a subsample loop to a laser spectroscopic N2O analyzer, both of which are measuring at a 1 Hertz frequency. The sample gas is then directed back to the chamber, forming a continuous loop. The analytical instruments were housed in 2 on-site trailers, both of which were connected to line power, ensuring near continuous operation. So a complete 8-chamber sampling sequence for each trailer repeats about every 3 hours. So for a total of 128 chamber flux measurements per day. Soil sensors that measure continuous soil temperature and moisture were also installed at every individual microplot in the soil surface within the top 0 to 10 centimeters. Half-hourly precipitation was also measured from a nearby rain gauge. So first we have the cover crop system on the microplots, and these were seeded in the fall of 2018, and flux measurements were conducted during the summer of 2019. So here we have 4 legume cover crop treatments with 4 replications each that were established on the 16 microplots. The treatments were winter pea, shown in blue, winter barley, shown in yellow, a 50-50 mix of winter pea and winter barley, shown in red, and fertilized winter barley, shown in gray. And I'm gonna color code these treatments the same way throughout the presentation. So the fertilized winter barley plots were established on half of the microplots and fertilized with potassium nitrate at a rate of 112 kilograms of nitrogen per hectare using a low pressure sprayer. The other half of the microplots remained unfertilized, and the data from these plots were not included in this study. At the same time that the gray fertilized winter barley plots were fertilized, these yellow unfertilized winter barley plots were sprayed with water to simulate a wetting effect. The crops were cut down mid-July, and the biomass was left on the plot, which is typical of an alternative crop rotation, adding nutrients back into the soil to increase soil health. So, and these circles here on each of the microplots show the locations of where the soil collars were installed between the crop rows. And so these are the spots where the N2O emissions were measured. About 3 months later, in the fall of 2019, winter wheat was seeded and fertilized by hand on all of the microplots, which is representative of a typical business as usual rotation for this region. In October, ammonium hydroxide fertilizer was applied to all the microplots at a rate of 168 kilograms of nitrogen per hectare using a banding application method. So each microplot had 3 bands of fertilizer applied, shown by these yellow dotted lines, and 3 seeded rows about ½ inch downslope of the fertilized rows, shown by the green lines. And so as the crop roots grow downwards, they will eventually reach the fertilized bands in the soil. The soil collars were left in place as to not disturb the soil, and you can see that this time the crop and fertilized rows were inside of the collar. The winter wheat system is still being monitored, but the data presented in this study is only up until February 29th of 2020. So here I've graphed the daily cumulative precipitation as well as the mean, maximum, and minimum daily temperature that was measured during our study to give you a full picture. So here is the cover crop study period, and here we have the winter wheat. The cover crop study was seeded back in the fall of 2018, like I've mentioned, and then fertilized at the end of May, a month before the start of a very hot and dry summer. And the measurements lasted through to the end of August. We only got about 50 millimeters of rain during the entire study period, and the daily mean temperatures ranged from 9 to 27 degrees Celsius. The fertilizer that was used was nitrate-based. So as a reminder, that's the substrate used by denitrification under waterlogged conditions. So makes more sense to use it during the summer. The winter wheat study was fertilized mid-October with ammonium fertilizer, so the substrate used by nitrification, the aerobic process that I had discussed. During the winter wheat system, October alone had 50 millimeters of rain, all of which occurred right after fertilization. So we had a really big rain event right here. And Flux measurements started right after fertilization and seeding and went through to the end of February. And the total precipitation measured during the study period was around 265 millimeters in the form of rain and snow, and the daily mean temperatures ranged from -9 to 13 degrees Celsius. So now let's take a look at the Flux results from the cover crop study. So this is what the cover crop study Flux data looks like plotted. The top 4 graphs here show the N2O fluxes from each of the 4 treatments, as I've color-coded right here, with 4 chambers per each treatment shown by the different colors as well. The y-axes are in units of grams of N2ON per hectare per day. And the vertical dotted line right here shows when the fertilized winter barley plots were fertilized, and at the same time when the unfertilized winter barley plots were sprayed with water. And the H shows when harvest occurred and the biomass was left on the study plots, like I had mentioned. This 5th plot right here shows the mean daily chamber temperature, ambient temperature, and soil surface temperature. The second to bottom graph shows the water-filled pore space percentage from all of the soil sensors at each of the microplots. And the last plot right here shows half-hourly precipitation measurements. So after each rain event, we observed that water-filled pore space sharply increased and then decreased quite quickly thereafter, suggesting rapidly draining soils and or rapid moisture evaporation due to the warm temperatures. So you can see some hot spots and hot moments that were near the beginning of the study, which were mainly the product of increased soil moisture from the few rain events that had occurred, likely causing that birch effect that I had mentioned. And so with this data, I analyzed individual N2O pulse events and their influences, or what influenced them, since it's kind of difficult to just take this all in at once. So in the next slide, I'm going to zoom in on this period of time right here to analyze mainly this hotspot pulse of N2O in more depth. So here's a close-up of those N2O fluxes sort of transposed horizontally in response to some of the soil variables that I mentioned in the background slides. So this right here is the highest pulse that we observed during the entire cover crop study in the winter P treatment. So we had 2 rain events shown by the orange and blue arrows, and the wetting of the winter barley plots and fertilization of the fertilized winter barley plots. So this first rain event shown by the orange arrow accumulated about 14 millimeters after a very long dry period. So this was likely a drying and re-wetting cycle, which likely triggered the rapid mineralization of soil organic matter, which in turn created ammonium substrate for nitrification, which then creates nitrate substrate for denitrification. The mean water-filled pore space across all microplats was at the transition point between nitrification and denitrification, so around 60%. The soils drained rapidly and didn't stay saturated for very long at all, so nitrification was likely the dominant process. But that said, even when nitrifying bacteria are very active within the well-aerated soil pores between soil aggregates, the aggregates themselves can become saturated, creating microsites for denitrifying bacteria to be very active. So this second rain event right here contributed about 8 millimeters more of rain, and water-filled pore space by that time had decreased to about 50%. So the largest N2O pulse observed during the study occurred right here at chamber 4, and it lasted about 10 days with a peak flux of 27 grams of N2O per hectare per day, approximately 70 times higher than pre and post background levels. So during this pulse, soil conditions were still moist from the first rain event, and the daily mean temperature was also steadily increasing, creating those optimal conditions for the microbial community to utilize the newly released nitrogen substrate, likely producing that hot spot and prolonged pulse of N2O emissions. And this second rain event commenced about 10 hours after the pulse began, potentially prolonging the pulse further, but not causing it. Besides the pulse, overall, the fertilized winter barley plots, or fluxes, were slightly higher than all of the other unfertilized treatments, but not by much. And since it was nitrate fertilizer that was applied, which is the substrate for denitrification, this was likely the microbial process that caused this overall increase. So here I've plotted N2O fluxes on the y-axis and water-filled pore space specific to each chamber on the x-axis during the entire cover crop study. And I've split this up by treatment as well. And the different colors show the different chambers. And also note the difference in scales for the y-axis. So you can see a slight positive correlation between the two variables, water-filled pore space and flux. During the study, mean water-filled pore space generally remained below 60%, which is the favorable range for nitrification. And just to point out again, here is that chamber 4 pulse occurring right around 40% water-filled pore space. And then just looking at the remainder of the cover crop study, we had another pulse and hot spot right here in the fertilized winter barley treatments. And following crop termination on July 10th, the combined mean water-filled pore space remained at around 16% until the conclusion of the cover crop study. So despite the fact that cover crop residue was left on the microplats after this point, the dry soil conditions were unfavorable for mineralization, as well as for both nitrification and denitrification, which likely explains the low N2O emissions that were observed for the rest of the study. So moving along to the winter wheat study. Here's a photo of our chambers during the winter. This is kind of how we would unbury them to keep them running. So you can see that on the y-axis, showing the N2O fluxes, they're overall much higher than the cover crop study, which peaked at 30 grams of N2O per hectare per day, but here we go up to 180. These graphs, again, are split up in the same way that they were during the cover crop study by the various treatments, even though during the winter wheat study, they're all fertilized. So during this study, we fertilized with an ammonium fertilizer, which is the substrate for nitrification. And so during this time between after we fertilized and before this wet period right here, Nitrifying bacteria likely got to work consuming the fertilizer and producing nitrate. However, winter wheat is planted and fertilized early, but doesn't come up until the spring of the following year. So that nitrate is potentially just sitting there in the ground. And then once we start getting into the wet season, rain, snow melt, and freeze-thaw events saturated the soil, creating optimal conditions for denitrifier bacteria, which could now consume the substrate, making these the main triggers of the N2O pulses during the winter wheat system, mainly between January and February, as you can see right here. So I'm going to zoom in again on this neat little portion of the data in the next slide. This gap right here in the Flux data is where we had to shut down our instruments prior to what I believe was the largest snowstorm that we had last winter between January 11th and the 16th. During this time, about 40 centimeters of snow had accumulated on our study site, which quickly melted just a few days after, causing this prolonged pulse event. So the fluxes I'm showing here are what we measured right after we removed enough snow from around the chambers and turned the instruments back on following the snowstorm. So between January 22nd and 29th, right after all that snowfall, shown here by the red parentheses, there were a series of daily light rain events, and the temperature also started to slowly increase each day, ultimately melting the entire snowpack about a week after it had accumulated. So between this time, the rain gauge measured about 92 millimeters of cumulative precipitation from the melting snow and the added rainfall. And so water-filled pore space reached levels of about 76 plus minus 8%, so well into the denitrification range. This black arrow right here indicates more snowfall, again followed by rain, for a cumulative precipitation amount of 58 millimeters between February 4th and 7th. And water-filled pore space was more or less maintained at the same levels. So these prolonged soil moisture conditions caused the largest and longest N2O pulses measured during the entire winter wheat study. And you can see some hotspot areas at chamber 1, shown by the blue, chamber 16, shown by the yellow as well. And interestingly, all 4 of the microplots that were previously under fertilized winter barley during the cover crop system had the lowest overall fluxes during the winter wheat system. And this was observed for the rest of the study period as well. So I'll touch on this later. Here again, I'm showing water-filled pore space versus fluxes during the winter wheat study, split up by treatment type. And color-coded by individual chambers. And the figure shows that the highest N2O fluxes occurred at a water-filled pore space of about 60%, shown here by the red dotted line. And similar relationships have been observed by several authors during that did field experiments in agricultural soils who found peak fluxes usually occurring between 60 to 80% water-filled pore space. So now let's see how N2O fluxes measured during the cover crop and winter wheat studies contributed to overall field scale cumulative emissions. So these 4 graphs show cumulative emissions split up by treatment, once again, and each of the 4 chambers in each treatment. So the scaled cumulative emissions were determined by using the daily mean flux values from each chamber, showed by the dotted lines, as well as the daily maximum and minimum. So at the bottom here, I've listed the mean cumulative emissions, as well as the standard deviation for each treatment by the end of the study. So all of this averaged to about 78 grams of N2ON per hectare for the entire cover crop system. The fertilized winter barley treatments had highest overall emissions, followed by unfertilized winter barley, then winter pea, and finally winter barley, or winter pea and the winter barley mix. This hot moment is the one that I did that little microanalysis on. And without this pulse, the cumulative emissions of that chamber would have amounted to about only 46 grams of N2O per hectare. But the pulse upped that to 110. So that resulted in 138% increase. So without this pulse, the emissions from this treatment would have been similar to those from the winter pea and winter barley treatment, the lowest overall emitting treatment. So this just goes to show how much hotspots and hot moments can disproportionately increase cumulative N2O emissions. So the mean cumulative emissions from all three of these unfertilized treatments were only about 34% lower than the cumulative emissions from the fertilized winter barley plots, suggesting that mineralization rates in the unfertilized plots were likely high enough throughout the study, particularly at the beginning, to produce similar quantities of reactive nitrogen to the fertilized winter barley plots. And subsequently, N2O production was similar through microbial nitrification and denitrification. And since N2O from the fertilized winter barley treatments is likely the main product of the reactive nitrogen created through biological fixation and mineralization, it could be presumed that the mean net N2O emissions from these treatments are background level for winter barley crops during the study. So subtracting that value from the cumulative emissions from the winter barley, the fertilized winter barley, that difference in nitrogen loss is likely due to that added fertilizer. And this resulted in about a 17% difference, which equates to about 0.014 kilograms of nitrogen per hectare, which is only about 0.01% of the original 112 kilograms of nitrogen that was added as fertilizer. So here are the cumulative N2O emissions from the fertilized winter wheat study. And you can see how they slowly started, or rapidly started increasing exponentially with the start of the wet season around December. Again, here are the total mean cumulative emissions for each separate treatment. And you can clearly see here that the cumulative emissions from the microplots that were previously fertilized winter barley during the cover crop study were consistently lower than all of the other microplots, the other treatments, and also exhibited much less variability. And so if we assume that all of the N2O emissions from this study were the result of N transformations solely from fertilizer, then 1.2 kilograms of nitrogen per hectare of the original 168 that was applied as fertilizer was lost to the atmosphere via N2O, or about 0.7%. And this value will continue to increase as the study continues through fall of 2020. And according to the IPCC Tier 1 emissions factor, 1% of nitrogen fertilizer inputs are emitted typically as N2O from agricultural soils. So now to synthesize all of my findings from both studies, N2O fluxes were higher from the winter wheat study compared to the cover crop study, which was explained by the fertilizer application location and timing. Fertilizer was only applied to 4 fertilized microplots during the cover crop study and not all of the microplots. And nitrogen fertilizer drives microbial activities, or nitrogen drives microbial activities that produce N2O. Timing of the fertilizer application also played a really large role in total N2O flux, because during the cover crop system, fertilizer was applied near the start of a very hot, dry summer. So there was just not enough water-filled pore space to stimulate microbial activities that produce N2O. Alternatively, fertilizer during the winter wheat system was applied right at the beginning of the wet winter season, which created those optimal conditions for the main N2O producing microbial process, which is denitrification. During both studies, water-filled pore space was found to have a slight positive correlation to N2O fluxes, with the highest fluxes occurring at around 60%. Hotspots and hot moments, or pulses, were observed during both studies. During the cover crop study, the main driver for the pulses was that drying and re-wetting birch effect. And during the winter wheat study, pulses were mainly caused by freeze thaw, snow melt, and rain. So now to compare how my results match up to IPCC Tier 1 emissions factor for agricultural soils and to another similar study to our study. So nitrogen fertilizer was observed to elevate overall N2O emissions from both studies. But again, if from the cover crop study, if we consider that the unfertilized winter barley plots as background N2O emissions and the N2O emissions from the fertilized winter barley were the emissions from the nitrogen fertilizer, those are greater by 17%. So attributing this excess nitrogen fertilizer loss, 0.01 was emitted as N2O, and this is much less than the suggested IPCC Tier 1 emission factor for agricultural systems, which again is 1%. And this also includes organic, so nitrogen from residue from organic matter is also added to the IPCC emission factor if you have that value. So during the winter wheat study, attributing all N2O loss to the nitrogen fertilizer, since every microplot was fertilized, 0.7% was emitted as N2ON. And this is close to that 1% value. However, keep in mind that we're only 4.5 months into our study. So by the completion of our study, our emission factor might be 3 to 4 times higher than this. And again, it also doesn't include crop residue nitrogen amounts. So Waldo et al. Studied N2O flux chamber emissions from an agricultural site at WSU Cook Farm under a conventional till and a no-till site from a fertilized spring canola between spring of 2014 to fall of 2015. So this timing is most similar to our cover crop study. Two of the main differences were that we seeded our cover crops the previous fall and fertilized the fertilized winter barley plots in the spring, while they both seeded and fertilized in the spring. They were also fertilized a month earlier than us, and that had, during that time, a couple of heavy precipitation events a month later triggered one large N2O pulse that they observed in their study. Whereas we were already into that dry season, but we still saw a few lower pulses. So overall, our cumulative emissions from the fertilized winter barley plots were much lower than what they had recorded, but our fertilized winter wheat plots were much more comparable and will likely end up being larger, just due to the fact that we will have had longer periods of saturated soil conditions. And our error value alone is 1.8 kilograms from some chambers. So it is already much larger from certain chambers, and we're only really a third of the way into the growing season. So now I just want to go over some potential future work and touch on some limitations of our study. So measurement of other soil variables throughout both the cover crop and winter wheat studies, such as soil pH, soil organic matter content, soil nitrogen from both natural and fertilized sources, could provide insight into how they impact soil microbial abundance and diversity, which would ultimately affect N2O emissions. Measuring variables like pH and microbial populations could maybe help us understand why the N2O emissions from the previous fertilized winter barley plots were the lowest that we saw during the winter wheat system. By measuring organic matter nitrogen, which is largely contributed to the soil via crop residue, we would know how much reactive nitrogen is being added through natural sources. And that could potentially mean decreasing synthetic nitrogen fertilizer inputs. Using stable nitrogen isotopes in fertilizers, specifically N15, to determine whether nitrification or denitrification were the dominant processes at any given time is also a method that is increasing in popularity. This was a limitation for me, just because I could only speculate what microbial processes were dominant at any given time. And a PhD student, Melissa Radanovic in Dave Evans's lab, is actually using the same study plot as mine and running experiments like that. So some of our fertilizer was tagged with an N15 isotope, and nitrogen isotope fractionation is dependent on the microbes processing it. And these different signatures can be analyzed via the N2O emissions by a spectrometer with isotopic measurement capabilities, which one of our analyzers had. So these findings will be available hopefully by the end of the year. Some other potential future work could include monitoring the effects of snowpack on N2O emissions. Since our study site was difficult to get to during the winter, we couldn't operate our chambers during the snowstorm event. And it would have been interesting, I think, to see how the insulating properties of snow could actually still be contributing moisture to the soil and driving N2O emissions. So here is a little snapshot into the surface soil temperature time series data. And we can see that this diurnal temperature variation essentially stops while the microplots were covered in snow. And water-filled pore space went up by about like 20% during this period, which put the soil in denitrification range. Some more future work could include increasing spatial coverage, which is a common limitation of automated chambers. Chambers are typically dedicated to the soil collar that they're installed on. And although it's beneficial to monitor how N2O fluctuates from the same soil surface location, spatial coverage is sacrificed. So 16 chambers can actually be hooked up to each multiplexer. For this study, we only had 8. And this is because we wanted to measure each chamber as frequently as possible to increase the temporal coverage. But to increase spatial coverage, more chambers can be added and the flux measurement period can be shortened. Eddy covariance can also be used to increase the spatial coverage by implementing the eddy covariance system, which is a common analytical method used to measure field scale CO2 fluxes. Of course, the issue with N2O is that it's emitted in very low concentrations from the soil. So the analyzer would need to be low enough to measure the fluxes, and this could potentially go against some of the major assumptions of eddy flux models. Either way, using flux chambers could potentially be used to ground truth the EC flux data. And so incorporating this analytical method could really help us capture that spatial temporal variability of the N2O emissions. And finally, we will continue monitoring N2O fluxes from the winter wheat study so that we can see the total N2O losses throughout the entirety of the growing season and hopefully develop an emission factor, since the IPCC Tier 1 emission factor isn't really representative for all regions and specific influencing variables. So I want to end off my presentation by thanking a huge number of people involved in this project. This probably isn't even half of them. I want to thank my committee for guiding and mentoring me through grad school and helping me with my research, and to Brian Lamb for being my mentor and introducing me to Shelley. I didn't exactly fit the mold of a typical grad student candidate, and you both took a chance on me, so I really want to thank you for that. And to all the people that helped with the extensive amount of field work, and all of the people involved in the Landscapes in Transition project, to NIFA for providing the funding for this project, and to all the PCFS employees for helping us with setup and site maintenance of our study site. So thank you, everyone.